Hello and welcome back. We're in conversation with West and Uli Lindauer, and we're going to be talking about Tesseract Life Sciences today. Specifically, we have a handful of questions that are all regarding the topic of spray drying for biologics, proteins, and advanced pharma. Will and Uli, if you guys want to give yourself even just a really brief description so that people can understand where your perspectives are going to be coming from in these answers, then that would be great.
Sure. I guess I'll jump in. Will West, co-founder and CEO of Tesseract Life Sciences. Tesseract has a number of operating subsidiaries, but essentially we have advanced delivery technologies and an integrated supply chain. It spans a number of domains, and today we'll focus on, in particular, our expertise in the peptide world.
Yep. Hi, everybody. Uli Lindauer, Chief Marketing Officer at Tesseract Life Sciences, and quite excited to talk about the topic today because I think that while it does have some technical angles, I think it's a very consumer centric topic in terms of what do biologicals do, how are they processed, what are they really? I think everybody's kind of wondering today. You see a lot in the press, you see a lot in the marketplace. So this is going to be great. And of course, we are going to focus the conversation on our subsidiary Advanced Powder Dynamics with their technology, and from there kind of, have the conversation how we do what we do and why we do it the way we do it.
Yeah, that's awesome. All right. Well, let's jump in straight to the first question, which I think is a great place for us to get started, which is why are biologics and protein-based therapeutics so challenging to convert into stable powder form, and how does that spray drying solve that?
Should I start with this one?
If you like, I can start and then you can jump in and correct me. That's also good.
I never correct you. You're usually spot on, so go for it.
All right. So I think there is two and a half answers to your question, Stephen, which is quite interesting. So biologics and protein-based therapeutics are very challenging to process and work with because they are incredibly fragile. They are not very pleased being surrounded by heat. I don't know, maybe you have heard the expression that a protein denatures, which is what happens when you cook proteins, for example, or heat proteins. We all know what happens to an egg when you heat it, when you cook it, fry it, stir it, or make an omelet from it. So it basically heat destroys the helix structure of a protein and splices it in many different directions, and therefore it is no longer intact. Biologics in general, again, have a very similar set of characteristics. They don't like moisture very much. It corrodes their molecular integrity. Same with the proteins. Some proteins are not terribly water sensitive, but many are very moisture sensitive. That's kind of why they are difficult to convert into stable powder form. Now, the half answer I have for you on the last part of the question, how does spray drying solve that? I will tell you it does not. And this is the most interesting piece of this question. Spray drying in the traditional spray drying form does not solve that. That's why most of these biologics and protein-based therapeutics or peptides are today freeze-dried, which is an extremely expensive process. Now, let me bring in our subsidiary, Advanced Powder Dynamics. Advanced Powder Dynamics has a process that looks like spray drying, but it is not. It does not provide elongated or long duration heat exposure on the molecules, which is great for biologics and protein-based therapeutics because they are heated for such a short period of time that they actually never really see the highest temperature that we have in our drying chamber. So it's an extremely gentle process. It is more flash dried than spray dried, and the end result virtually looks like spray dried material. very fine flowing powder, very easy to work with. If we apply our cyclodextrin inclusion, it is extremely soluble, which is also very terrific. It protects the integrity of the protein. One of the most interesting pieces of the array of services that advanced powder dynamics offers is, among others, the ability to dry egg whites and egg yolks in their original molecular protein shape, meaning no denaturation happens. The actual protein chain of amino acids is not destroyed, it's not compromised, and it can be reconstituted without any loss of nutritional value. And that's why we are firmly in the camp that this process that APD deploys, dynamic atomization, is probably with freeze drying the single only version of creating stable, easy to work with, very durable powders in that particular area of biologics.
Hmm. Yeah, it's funny. Even just from our other conversations that we've had about APD and some of the questions that I've gotten to hear you guys and Hendrik answer, when you were describing what the difficulties were with that, I immediately thought, wow, the whole dynamic atomization process that you guys described that the APD utilizes seems like the perfect solution for the problems that arise when you're spray drying these types of proteins.
As usual, Uli's spot on, but I love the way the question is posed as if there's only one biologic profile or only one protein profile, and therefore an answer can be applied to biologics as a class or proteins as a class, because the reality is that they're categories, but within them there are literally thousands of iterations. This is what's fantastic about our line of work, our business. People are coming to us from all over the world to make a better version of the peptides that are becoming so popular right now, just as they have in the past, to make better versions of ingredients in the natural product space or better versions of OTCs and so on, because we don't manufacture peptides, but we take the absolute best products that we can get from the manufacturers, and we apply our extraordinary delivery technology to bring out the best of them, to make them more bioavailable, better absorbed, etc. I was making a couple notes as Uli gave her explanation, and I'll try to keep this very brief, because what we're here to do is try to make information accessible to an audience and ask her and answer very important questions, but in a simple way before we dive into a lot of the details. And the list that I was making is, what are the things that are on people's minds right now? And what are on their minds right now are many of the peptides that people are going out and buying. Whether they should or not, that's another question. Everybody has a different opinion about that. But things like BPC 157, CMAX, KPV, TB 500, there's something called the Wolverine stack, for example, which has a series of peptide stack within it that's incredibly popular. Others, Kispeptin, melanotan, CJC 1295. These are things that people are running businesses all over the world coming to us, asking us to upgrade in their bioavailability and performance. And it's because the momentum, the velocity at the consumer and the business and wholesale level is growing very, very rapidly. It's exactly why the FDA is taking quick action to regulate these peptides and how they're produced, how they're managed, how they move through sales channels, where they become available through doctors to patients, to the consumer market, etc. Very wisely, I would add, but it's changing very quickly. All of them, however, are very different. Some are quite small at a molecular level, which means that they behave differently when you try to dry them, when you try to turn them into powder form from injectable form, liquid form. And that means that they're going to have a different outcome, regardless of what technology that you use. So Uli's first point, they're fragile. They're absolutely very fragile. That's true. Their fragility changes depending on their size and their structure. Their structure is the second point. They become very complex molecules. And the ability to deal with them changes as they grow. The more complex they are, the harder they are to manage, and the more difficult it is to improve their performance, and also simply to dry them and to make them manageable, to make them sophisticated yet accessible to the human body for absorption, for bioavailability. And not every drying technology, absorption technology, etc., will work for them. So this is one of the reasons why advanced technologies and specialties like the ones we try to advance every day become so important as this portfolio of peptides continues to expand for the medical and the consumer markets. It's interesting because people are learning about peptides very quickly, but right now we actually have quite a small portfolio of peptides available to the world. But every day it grows, and in a year or two we'll have twice as many, and a year or two after that we'll have twice as many again peptides available for medicinal and other reasons, which will serve the markets. But it'll be far more challenging to dry those peptides, to advance their capabilities and make them accessible and bioavailable to the body. So it's fragility, but it's also complexity that drives what we can do with them and how we do that. The final point is really about that solubility. Right now there's a big push to make these injectable peptides oral and put them in powder form. And that's a challenge that will reach its limitations, because moving things from injectable to oral form certainly reaches barriers for what you can do with the technology to make that translation, but also there's barriers in the human body. The human body simply will not absorb certain things above a certain size. And so we will very quickly reach limits there, regardless of which technologies we're able to use.
That's interesting. I think you brought up so many good points in there that that I think actually you're going to end up expanding on in a handful of these next questions. One of which I think you started to talk specifically a lot about the size and the differentiation within that size, which is I think what this question really is getting at the heart of. So what's the difference between spray drying a small molecule pharmaceutical versus a biologic like a protein or peptide? And what changes specifically in that process?
The differences exist across the entire spectrum, starting with the supply chain going on to, I'm talking about in the corporate world as well as the technology world, and then the markets themselves. There are differences in the supply chain and how they have to be sourced. There are differences in processing and how they have to be handled and treated. There's differences in the technology, which Uli alluded to. There's certainly differences in the delivery capabilities and the intellectual property that exists behind those hardware and delivery technologies. There are massive differences in stability or lack thereof that are tied to those points that I made: the size, the fragility, what you can do with them as you process them. Massive differences in regulation and regulatory approach, which are starting to change now and affect consumers and the health channels and so on. So taking those in order, naturally from a supply chain point of view, people can respect the fact that peptides are very sophisticated molecules, and the vast majority of them are manufactured, synthesized. They may exist, some, in the human body, but in order to reach the markets as they are now, they have to be produced in a factory or a sophisticated lab. And that drives up the cost, makes them very expensive, and it makes for very expensive handling of those. We started off our business in the supplement, nutraceutical, and medical space categories, and at times when we had to buy a product that was $1,000 a kilogram or in that range, we would recoil in shock and fear and sometimes stay away from it because it was so expensive to produce, a bottle of 60 or 120 capsules when your ingredients cost that much money. Now, peptides have prices that are hundreds of times as expensive when you're buying them at scale. And so it's orders and orders of magnitudes more expensive to manufacture or to purchase peptides just because of the way they're produced and what they are and what they represent and what they can do. And that will evolve over time, but it's going to be very, very slow. The handling of these peptides is very important. As Uli mentioned, they're quite fragile. They have a very short shelf life. Typically, these peptides are lyophilized or freeze-dried when they're manufactured. That gives them a shelf life. It can be 60 days, 90 days, can be longer, but only if they're stored at very, very cold temperatures to help preserve them. When they're reconstituted, that starts a ticking clock, and typically they have to be used within weeks. Rule of thumb is that most are good, for a month, give or take, once they're back in liquid form, but they have to be consumed pretty quickly. And that's a problem when you have an extremely expensive product and a very short shelf life. It's a very big challenge for the supply chain. So they have to be transported cold, used quickly, and it's very expensive for a business if they go bad. So there's a big push, therefore, to find ways to extend the shelf life with a lot of technology. And that's one of the things that we've put a lot of emphasis on, is how to use our capabilities to dramatically extend that shelf life with things like our core expertise in cyclodextrin inclusions and so on, where with our other lines of business, we can extend shelf life to periods of years, with our other products. And so we work to do that now with peptides as well. For the consumers or the patients who use them, that obviously adds value to them as well. When it comes to delivery systems, then the translation to oral is similar. You want to find ways to deliver the peptides in the body in oral form. There's obviously going to be a push to do nasal sprays and other things as well, which is already happening. But whether you're talking about oral delivery or injectables, etc., the point is bioavailability, and whether or not you have technologies that can really radically improve that. We're fortunate that we have multiple tools in our arsenal, an advanced delivery system that can improve the translation from injectable to oral in ways that appears to be 5x the improvement that you see with things like liposomal and other tools. As we've seen in past businesses, we have technology that the pharma industry doesn't have in sodium caprate, and the ability to magnify absorption that no one else has. And we have strategies for changing other aspects of the translation in our drying technology that no one else has, that we've talked about before at the APD and manufacturing level, where in our cyclodextrin inclusions we can eliminate water activity. We can create a freeze-dried quality powder that has the improvement of the throughput of lyophilization, so we can manufacture quicker and better than lyophilization using a different approach. And yet that powder is highly water soluble, can actually be blended with lyophilized powder from other sources and stacked in vials for reconstitution. So it's a very innovative approach to all stages of this. But the important thing, I think, in this industry, because it's evolving so quickly and nobody knows from one week to the next what's happening, is versatility, both versatility in terms of products in the tool set, versatility in terms of the applications and market segments that can be pursued, and the versatility in ways that the different pieces of intellectual property can be deployed across the various peptides, regardless of their size, complexity, structure, fragility, or necessary shelf life in various applications. And so it's very need for a multi-dimensional approach, and it's challenging to find somebody who has, even one or two of those axes covered, let alone multiple axes.
So within all of what you just said, obviously there's a lot of different pullouts. I thought one interesting aspect that I want to kind of make a clarifying question on, because I think it'll be really helpful to people watching. So the inclusion of cyclodextrin could obviously create higher shelf life, higher solubility, higher bioavailability within the ways that you guys are actually utilizing them in the manufacturing process here, specifically with peptides. And so a peptide that has a cyclodextrin inclusion could significantly improve its actual usage to the consumer. Does that exist on the market yet? Or is that something that is only really able to be done through the specific kind of manufacturing that you guys are kind of frontiering currently?
Julie, I'll let you take this one.
All right. So when you include a molecule in a cyclodextrin cavity, there's a couple of factors that are very important, particularly when we talk about peptides. So peptides come in all shapes and sizes. I mean, I'm just going to throw out a couple of technical terms here. So a peptide can probably be anywhere from 60 Dalton mole weight to 130,000. Yeah, that is a very wide span. So a glutathione molecule is probably around 260, and then a clotho peptide molecule is 130,000 Dalton. So you can already see that there is no single answer. What I can tell you is that any kind of cyclodextrin inclusion, be it the approach that we usually deploy for our nutraceuticals, where an entire molecule is encased in a cyclodextrin cavity, or an approach that is more similar to what the pharmaceutical industry has been doing, where large molecules are capped on their branches with several, sometimes even different sized cyclodextrin molecules, dramatically enhances the solubility, protects the peptides on their way to their destination, and then really in combination with our sodium caprate has the ability to really freely and smoothly enter through the digestive tract if consumed in oral form, or through the blood and organ barrier if used as a reconstituted injectable. And so it is when you look at bioavailability on its own, regardless of absorption, timing, location, just look at the variable bioavailability. Cyclodextrin inclusion has been proven to multiply a molecule's natural bioavailability to the human body by about factor 4 or 5. So then that's already pretty dramatic, especially when you have something like a peptide that might only have a bioavailability of 10 or 15%. Now you think about what happens, you multiply that by 4 or 5, you all of a sudden have something quite enormous. And then, of course, in the chain of our technology, now you have the bioavailability created, but now you need to make sure that you have the timing correct, where it actually deploys this bioavailability and the absorption to make sure that it actually can get to the exact location where it is aimed at. And so I think the nutshell answer to your question is we are not aware of anyone else who's doing it with cyclodextrin inclusion. Yes, cyclodextrin inclusion is a tremendous unlock of bioavailability, but it has to be paired with the toolkit that we have, and that is a large part of our patent suite, to really get to peptides to an unprecedented efficacy.
Wow, that's exciting, honestly. So we'll move on to the next question here, which is what are spray-dried dispersions and why are pharma companies increasingly using them to improve bioavailability?
Basically, it's spray drying dispersion is an approach to spray drying where the maker is trying to avoid crystallization and improve solubility. And so they're dissolving material in a solvent, and then they're flash drying it, and there's a polymer involved. And so when they flash dry it, they're dissolving the material and evaporating the solvent, and the core material is left in a polymer, which really, I think what people need to understand are the two key drawbacks. One, it doesn't necessarily hold. Things can revert into crystalline form, which is counterproductive. That's what you're trying to avoid. Sometimes it works, sometimes it slides back into the undesirable crystalline form. The other things, though, are that one, you've got a solvent involved and a polymer involved, and that's not necessarily what you want. A lot of people don't really want or need polymers in their material, and yet they end up with one. More importantly, the spray drying itself is usually part of the problem. I mean, we've talked about this at length, so I don't want to spend a lot of time on it, but the spray drying itself is an inexact science. It's certainly imperfect compared to the dynamic atomization alternative. And when you compound its inefficiencies, such as the residual water, the residual moisture content, and water activity that can lead to mold and APC and so on over time, having these other drawbacks to the dispersions, it just adds to some of the challenges that you have to deal with. Now, it can be very cheap, and that's why people pursue it. There's a reason why people do things. So clearly, it's a decision that part of the market makes and will continue to make for reasons. If they're looking purely at economics, short term, don't care about the drawbacks. Not arguing with that, but it's important for people to understand why those decisions are made, despite the drawbacks. It's usually purely economics and a lack of concern about the purity or the efficacy of the final product. It's not the kind of approach that's taken for a very high-end, very sophisticated product. And so it's just on a spectrum at a very different part of the spectrum.
That makes a lot of sense. If the whole description, I think, of the beginning kind of context of the question, I think helped quite a bit with the understanding of the economics behind it and everything. And it's not surprising. It seems like as this industry grows, it doesn't shock me that that there are certain companies that are going to decide to take the lower end kind of pathway to make a cheaper product, while some of the people, seems like what you guys are doing, are making a much higher quality, much more pure and bioavailable product.
Well,
I think that's an interesting comment, and I do think that to some extent that gets you even to your next question, how dynamic atomization benefits sensitive biological materials. One of the things that I want to get us back to, where the economics become extremely important, is what Will was talking about earlier in terms of the raw material pricing. There are peptides that cost $70,000 a gram. And so when you look at that, what you really, if you are a company who's buying peptides like that, and maybe if you buy a kilogram, the price goes to $50,000 a gram. There are a few things you cannot afford. You cannot afford to lose really any in the processing, and you cannot afford to lose any to forces that you find in spray drying, like moisture activity or mechanical shear, right? So we talked about how fragile and sensitive these molecules can be. And in spray drying, oftentimes the material is pushed through the process with an incredible amount of pressure, which on a molecular level causes the molecules to rub against them and bounce off of each other, and then they're in the heat for maybe 20 seconds and they bounce off of each other, and in the end they no longer look like this, they kind of start looking like that, because it's just physical mechanical forces with which they bump against each other. And that's why, one of the aspects of dynamic atomization is that it's a very, I don't want to say gentle, because it doesn't feel like there is anything gentle about, heating a substance to over 1,000 Fahrenheit for, even if it's for less than 0.4 seconds, but it is it's a low pressure process, which is very beneficial. And that's why I think, our companies are uniquely positioned to work with those specifically challenging materials. We were out in the manufacturing facility yesterday and had a whole group of absolutely lovely, highly intelligent, very influential industry participants with us. And, one of the things that we were talking about is that in many instances, we are being approached to provide the solution to a problem that doesn't seem to have a solution. And so I think that's goes kind of into that direction. Like, why is dynamic atomization particularly suited? And the answer is yes, because it is especially well designed and engineered for particle engineering fragile materials.
That's what you said makes all of that make sense, but that is, I think, even just the very beginning of that to help people understand just how expensive some of these peptides are and how pure and precious they need to be basically
It's
treated.
crazy.
It's
It's
ins
crazy.
Yeah. No,
Yeah, I think
that's really
people
interesting.
people don't really understand. They understand. That peptides at the final phase, point of fail, they're quite expensive, but they're in many cases only buying micrograms per dose. And of course, at the level we're, participating in the value chain, which is at the beginning of the value chain, working with manufacturers to make their products in bulk, in mass, better before they flow through the value chain. We're dealing with, very large volumes of kilograms, and then they're being sold, bless you, being sold for hundreds of thousands of dollars per kilogram. That's basically the friends, family, and hostage price, because right now that's the stage the market is in. There are very few ultra high quality producers. There is an Asian supply component, there's a Middle East supply component, and there's a very, very nascent US supply component. But the value and the supply chains are still taking shape. And it's extremely important that anyone who's participating in this market have the materials rigorously tested to make sure they're extremely high quality. And in order to get the best high quality product, it's extraordinarily expensive and will be for a long time. So for now, until more supply is built up to meet the exploding demand, there will continue to be excessively high prices for pure product at the beginning of the value chain that will continue to flow through, and will keep these products at a very high price, even as the FDA begins to route particular peptides into the compound pharmacy channel, because demand for them seems to continue to grow as fast or faster than they can be produced at the origin of the supply chain.
And that's really cool to hear as more of an outsider from the industry because, you know, if a supplier is paying $70,000 per gram of some of these really, really high value peptides, and they're going through the processing of that, they have to charge a certain price to the consumer for the end product of it, whether or not it's been processed in a way that's very bioavailable or not. And so I think what's cool about what you guys are doing is you guys are adding a very specific connection within that value chain process where a company has to charge a certain price for an end goal product anyways, but the quality that the actual consumer is going to get out of that is a big factor. And you guys are adding such a big value aspect within that that I think not only is great for the consumer and different people who are on a more wholesale level going to be buying the product, but to the end consumer who's actually going to be consuming this or putting this in their body. And I think that that's a really amazing thing that it seems like your company is doing.
Yeah, that's what we're trying to do. Is we're trying to take phenomenal products that do amazing things for people
Mm-hmm.
and make them even more impactful and high quality, for them, for the value chain. What we see is that these products do have an impact. That's one of the main reasons people are seeking them out en masse, and it's growing so quickly. And what we're able to do is apply our technology layer to that, so that when people see the Tesseract name associated with the products that we've influenced with our technology, they'll know that they're getting the absolute best, most impactful, most bioavailable, and well-absorbed version of those products. And that's why we're very selective about the partners with whom we work, the original manufacturers, and the partners with whom we work downstream, who take our proprietary powders, our proprietary ingredients, where we've reacted these peptides and then incorporate them into their own products, regardless of what that product is, whether it's a powder-filled capsule or it's a gummy or it's another product for the value chain.
That's really cool. So I do have another question that I think is very pertinent to. We've talked so much about how this kind of breaks down on a bigger level and how this works out kind of on a theoretical aspect of different inclusions and different practices and things. But can you walk through a real example? Take out the name of the client or anything if you need to, but of a biologic product that you successfully converted to a stable.
Yeah, I'll give you two very quickly because I think they're very telling, and I think they're very pertinent examples. Choose them, for this particular market segment. I mean, there are a lot of them out there. The things that we're doing, right now for the peptide sector, all of those with partners are under NDA. But I think these things are relevant to that sector. They're items that are going into these products, but I'll speak about them, generically. The first one is a short chain fatty acid. It's butyrate. It's a product that is very relevant to formulations for gut health, and there are a number of products that are coming together for gut health in the industry, and Uli's involved in a lot of the work that goes into our partners in the peptide space that's emerging now, and that they're showing very strong interest not only in our butyrate, but a particular stack that we've done with butyrate, which is 2 molecules in one cyclodextrin cavity. It's butyrate and N-acetylcysteine stacked together. It's one of the patents we have is around stacking 2 molecules or more in one cyclodextrin cavity. Tesseract was the first to do that and show the synergies that we can extract from multiple molecules in one cyclodextrin cavity. But the butyrate molecule itself is extraordinarily volatile. Butyrate is one of 3 key short chain fatty acids, so you produce that yourself in your gut when you're breaking down fiber. And that particular biologic drives more than 300 key processes in the body. But because of its volatility, that raw material oxidizes extremely rapidly. And so it has almost no shelf life. What we've shown in our stability testing is that we can actually make that product last for years. The potency testing on that product actually far exceeds the 3-year shelf life that we put on our finished powder. So that's one example where we've taken a product that has virtually no shelf life, and I literally mean 3 months or less in raw material form, to something that has years of potency sustained once it's reacted with our technology. The second one is actually one of the original peptides. It's become quite well known and quite popular, which is glutathione. I think Uli's smiling because it's one of her favorite ingredients. Glutathione is something that is produced by each person's liver in small amounts. I wish we all produced more of it. The more we produce, the more we can slow the aging process, because glutathione neutralizes toxins in the body. It seeks out reactive oxygen species. It attaches to free radicals and prevents them from doing damage in the body because they attack other items in tissue and destroy tissue. And so if we had more of it, we could counteract that damage more aggressively in the body. It's one of the original, it's a tripeptide. And so what's also interesting about this particular structure is that we have amazing data to show particularly how powerful it is with our delivery technology. In its raw material form, it's unabsorbable by the body. If you take glutathione in oral form, it's fortunate for a person to move the needle 1 or 2%. That's about all the benefit you get in blood plasma readings. over time. Liposomal, by comparison, shows an improvement of, on average, 11 to 14%. And liposomal's not a bad delivery approach, all things considered. Our technology, on the other hand, in clinical study of 32 patients, showed that the average improvement was in the high 60s%, and the patients at the top end of the study were over 200% improvement in blood plasma levels. So all things considered, we have shown in that case that we can also achieve a 3-year shelf life with a fragile peptide, and we can get amazing delivery results in blood plasma levels, with the structure. So those are just 2 examples that we have deeper data on because of the benefit of time, but we're looking forward to the same results over time as we get more and more structures from biologics into the delivery system.
So just to truncate that down into a single statement, because that's so impressive. So glutathione, one of the original peptides that started to get used in this way, in a normal sense, without liposomal or cyclodextrin inclusion, is at a 1 to 2% raise would be good. Liposomal is, you said, somewhere in the region of 10 or 11%, somewhere in there?
11 to 14 is what
11 to
studies will
14%.
generally show.
And then with cyclodextrin inclusion, you're looking at, after an actual study being done, clinical study, at over 60%, with some people as high as 200%.
With our version, including our release technology, yes.
That is wild to even understand.
It is.
I don't even
Yeah.
understand that.
It is.
Wow.
Now, I would say that the people who should be most excited by that, Stephen, are the people that like great skin, great hair, maybe like to have a couple of drinks because the glutathione is great for those uses. It's often used in cosmetic applications and so on. Lots of other things too. But it's good news for people who understand glutathione and its benefits for sure. But of course, my second point is that our delivery approach can be applied to hundreds of different molecules already. And as we have more time, we'll apply it to hundreds more.
Yeah, no, that is those exact 2 examples I think were super, very, very useful in that obviously you have something that's not necessarily a peptide, which is what we're talking about, but also a peptide to just show the versatility of how this breaks down.
That's right.
So for the next question we have, what trends are you seeing in the biologics space? And I'll back that up actually. What trends are you seeing in the biologics space that are driving more companies towards spray drying as a solution?
Yeah, I think there are quite a few, and I don't want to cut Uli off, but so she can jump in. But I think there's, the key ones are, first of all, as I mentioned, these peptides are growing in number. They're growing in cost. So those are 2. They are delicate, which means they're expensive, hard to handle, require a cold chain. So people are trying to get away from the cold chain, and therefore they need stability. So there's a need to handle a high value product, but in a way that doesn't require special handling, right? So there's an irony there. And finally, I would say that people are looking to make them more versatile. Right now, if you want peptides and you want to take advantage of them, you get a vial and a needle and you inject yourself. And there's a lot of people who want to get away from that, and a lot of people who are interested in trying them as soon as other options are available. And so I think Tesseract's mantra internally at this stage is knowing that what we're working toward is being the company that says, we're going to give you what you want, however you want it. Do you want things in oral form? Great. We're going to make peptides available in oral form. You want them in another form? We're going to adapt them, translate them to another form. Do you want your peptides in a powder-filled sleeve so you can make a drink? We're going to make that happen. Do you want to make it more bioavailable? Check, we've already done that. Do you want to take those peptides and that already great bioavailability and condition your body to absorb it even more? Great, check, we've already done that. And then filling out that matrix of all these different axes so that it can be handled without cold chain, it has longer shelf life, it can be deployed across all these applications and all these market segments. We are making things versatile across all those axes. That's what we're doing.
It's interesting that you bring up that specific perspective of outcomes to the consumer and some of the aspects that you brought up because, and correct me if I'm wrong here, but it also seems like a lot of the companies that are maybe interested in getting involved in something like this, in offering a product that's of this nature, are likely offering other products that they're used to being able to spray dry because they're not necessarily as particular or fragile, so to speak, as these peptides. So it seems like also one of the values that you guys are bringing to the table is also making it so that some of the companies that are looking to get into the peptide space as an offering, you're also allowing them to keep a similar process to what they're already used to in the manufacturing of their other products, whereas otherwise they may have to actually completely look at a different option than what they're used to. Is that correct to think?
Well, that's very perceptive, and I think Uli can talk about this. But it's interesting that you pick up on that because we haven't actually said that yet. We haven't told you that yet. So clearly something's getting through, or maybe you've been talking to Uli and I haven't been around, but that's exactly it. I mean, people can move forward into the future with a very powerful new product line to take advantage of these consumer trends, the wave that's coming, capture the revenue, capture the margin, serve their existing customer base while attracting new ones, which they very much want to do without disrupting what they already have. Maybe they embrace a couple of new partners in that supply chain that they've already built. But the reality is that we help them keep business as normal, but just drop in a couple new building blocks before growing like mad.
And I think that's what's most exciting to me because when you were talking about that being the way that you're serving the end consumer of being able to offer them what they want, how they want it, that was originally where my thought went into because I think it's exciting that some of these really amazing brands that are already offering great nutraceuticals and great nutrients and different vitamins and other sources in the space can essentially, without changing their supply chain and their general way of doing business or their understanding of product costs or anything like that too drastically, they can pretty easily get into this space by utilizing the services that you guys are actually frontiering currently, which is awesome because more of those brands, there's some amazing ones already out there that have a great consumer base that would love to be able to offer peptides, and it feels like this is the most turnkey solution to that.
Well, that's exactly it. I mean, people have an appetite to participate, I think, and frankly, they'd be foolish not to. But at the same time, an appetite that's met with either fear or confusion or just wondering where do they start because it can be overwhelming. There's a lot of technology at play. There are a lot of questions. Anytime you're starting a new line of business or something like that, there's going to be doubts about how hard it's going to be, all the things that you don't know, and the things that you don't know. They add up. It's a long list. But here, it's really as simple as one phone call.
That's awesome. So moving on to the next, peptides are one of the fastest growing categories, as we've said, in pharma currently. What unique challenges do peptides present for powder processing and how are you approaching them? I feel like we've kind of quite a bit already, but I guess if you had to really summarize a lot of what we've spoken about into this direct question, as simply as you could, I guess how would you put that?
Well, I think there are a couple things, and I don't want to do all the talking, so Uli, you can tell me if you want to start or interrupt here. But was that a yes, you're going to interrupt, or yes, I should keep talking?
No, no, please do keep going.
Okay, yeah, she's always curious what I'm going to say. See if I put my foot in my mouth too. It's interesting, Stephen. One of the things I find comical is that part of the challenge here is the absolutely tiny dosing, and it's a real issue. But I also address it tongue in cheek. But I think people may find this interesting. We have to create a processed powder that's easy for people to work with downstream. And ultimately, people are going to be working with powders, which in some cases they're microgram doses. And it's important that people not take too large of a dose. We have to make it easy for them, which means we have to make it easy for our partners to make it easy for their customers who are the end users. And when the doses are so small, the value chain, meaning the supply chain, has to find it easy to measure and segment, the doses along the way. And so it's a real challenge to work with powders that are so highly concentrated in their potency, their efficacy, that when it comes time to segment dosing, you have to be sure that you're not accidentally overdosing. And so blending down the powders is important and a challenge. And when you're doing things in bulk that then goes downstream to any number of partners and any number of geographies and so on, again, for a number of applications, number of market segments, bunch of different uses, keeping in mind that we're at the beginning of the value chain, serving lots of partners, lots of applications, lots of geographies. So literally it's a myriad of permutations. It can be a challenge. If we were just doing an injectable product with a giant vat of liquid and all we were doing was dumping it into small bottles, dispensing it in small bottles, we wouldn't have to worry about this, because the ultimate dosing is somebody sticking a needle in a vial and pulling out, 2 milliliters or 3 milliliters or whatever it is. It gets measured individually by a doctor or patient or somebody, that way. It's a little different. But when you're dealing with powders, it's a funny problem to have, but it's a real issue that you have to think about. But ultimately, the bigger picture and the serious topic is the ultra fragility of the product, the ability to make it robust and a strong, resilient product that is sustainable on the shelf in a complex environment that will serve all these markets. And the translation from that liquid form into usually an oral form, which is what people want right now, that can serve all of these markets. If we can do that, then we can serve the entire channel and all of these partners around the globe in doing so, because we give them something that they can work with no matter what they're trying to accomplish, no matter who their partners are. And so the way we do that is with our core technology platform, which allows us to convert a liquid into a powder with this phenomenal manufacturing and drying platform that we have at Advanced Powder Dynamics, the unique way of capturing things in a cyclodextrin, excuse me, that gives them amazing shelf life, and then using our other intellectual property, recapture that molecule when we take it out of the cyclodextrin and put it into any number of market segments, in any number of form factors.
Yeah, I just want to add one quick thing. I think that, Will already talked about the fact that they're extremely fragile, they're extremely expensive, which is, the perfect storm. They have to be easy to work with, and we absolutely have to make sure that we can maximize the effectiveness and efficacy of the actual peptide for the consumer. Because while the peptides are, we talked about $70,000 a gram and stuff like that, to some extent, this cost is passed down to the consumer. And nobody really wants to pay, $200 or $300 or $400 for a pill bottle or an injection and not see an effect. I mean, that is really the last thing you want to do, particularly because, as with all things that go into the body, eventually they're metabolized. So you're not going to just do them once. You're going to continue using them on a weekly, on a monthly, on a daily basis. The other thing that I think is very different about peptides is that they have to be processed in a clean room rather than in a regulated good manufacturing processes, GMP, or food grade GMP environment,
Hmm.
just because the dosages are so small and you really need to avoid any kind of contamination with dust, swirling particles. So there's airlocks involved. There's a quite an elaborate airflow architecture involved, so it is several steps up from, let me call it, regular supplement manufacturing, which again, adds a level of protection for the consumer, protection for the channel, obviously protection for the peptides themselves and how they're being processed to make sure that they maintain the highest possible integrity.
That is really interesting that they have to take even a further level of precision to be handled
Mm-hmm.
correctly.
Yeah. Well, because they're just in micrograms, right? They're fragile, they're small, and you only give like 300 micrograms, which is literally 300ths of a milligram. Like that is like how
No, yeah. That's I think that the majority of people probably have a hard time even understanding the a minuscule aspect of that. So that's really interesting. To be honest, I almost borderline expected the answer to that question to be similar to a bunch of the other questions that we've asked before, but I think that was honestly such a unique answer that I didn't fully expect with it. So, moving on to the next one. How do you measure and verify that a biologic maintains its activity and structural integrity after going through this drying process or the way that you process it?
Yeah, in this respect, this part of our business is like the other parts. I mean, you can look at these forms under an electron microscope, for example, but most of the analysis is sophisticated, traditional lab analysis. You want to look for potency and understand that the material has survived the reaction, survived the heat of the drying, and so on. And then ultimately, the biggest test is the consumer test. You'll see the products perform in the market, but the traditional lab analysis is really the same. There are labs that specialize in in the peptide analysis, and you'll see more of those over time, serving the manufacturers, serving the value chain, serving the compound pharmacies that are coming into the picture as we go forward under the FDA emerging FDA guidelines, and they're using all the usual analytical techniques that the labs have used historically in pharma and in the supplement nutraceutical space.
That's interesting. And so one thing that I think came up with this in our questions, we've talked a lot about specifically how stabilization can be affected via cyclodextrin inclusions, but how does cyclodextrin inclusion play a role in stability in comparison to other methods like lyoprotectants?
I'd like to take a stab at this here because so when you look up what lyoprotectants really are and what they are made of, so they are actually used to protect the peptides particularly during lyophilization, which is the same as freeze drying. You read things like there are polyols and polymers, and I'm like, I don't want that in a product that I take. I really don't. I mean, I know there's a huge debate on microplastics right now and the accumulation of microplastics in the body. And I'm sure not every polymer is a microplastic, but it's nothing that feels particularly conducive to be part of a substance that I would voluntarily take as a consumer, be it as an injectable or be it in an oral form. And so cyclodextrin inclusion plays a big role for the stabilization, but it plays an even bigger role in protecting the peptide on its way to its target destination, right? So think about it as a little submarine shell that goes around every single peptide, protecting it from degradation while swallowing in the esophagus, stomach acid, all sorts of things that can happen to it on the way into your, if taken orally, on its way into your large or small intestine. And so I think that is the bigger role that the cyclodextrins play here, because we talked about it earlier. They can increase bioavailability of a substance by 4 to 5 times, and so it's really the best of both worlds. It's a starch derivative, so it's actually not plastic, or it is. It's basically, cyclodextrins are prebiotics, and so in a strange way, because they're carbohydrate derived, right? So in a strange way, it offers so many advantages over lyoprotectins. We've been talking about them for a really long time, but I'm sure, Will, you have to add something probably.
Oh, I think that was a pretty good explanation. Yeah, I think that
Yeah.
in general, the lyoprotectants are just less stable, which is the general theme here. And when you have the extraordinary stability that the cyclodextrin deliveries can provide, and then you have the opportunity to enhance that with our unique patented capabilities to unwrap the cyclodextrin and pull the molecules out literally one by the billions and present them in the body for absorption. Why would anyone pass that up? I mean, that's a unique opportunity. So, again, going back to an earlier theme, people choose different delivery approaches, they choose different drying approaches, they choose different things for different reasons, but usually they come back to an intersection of a certain price point and a use case. And we just happen to be at one end of a spectrum that's really suited for premium ingredients, premium applications, and optimum delivery, because we can do things that are, I would say, atypical is an understatement. And so when things really need to be optimized, the cyclodextrin delivery that is perfected with our intellectual property and the things around it, they almost always make the most sense.
That's a really kind of interesting explanation from specifically from going back to the original thing that you said, Uli, and in going back to really looking up what they are and the fact that more closely related to that of a polymer, whereas a cyclodextrin is more closely related to something much more similar, it seems like, to almost not
Food.
necessarily a food, but so to speak, something that looks much more similar to what we would experience from other food products. And it's just interesting to me that that is a common usage in the industry where you're directly serving people who are so acutely and precisely trying to get a product in their body that is so specifically made for optimal health that you would end up, I don't want to say sacrificing, but
Compromising, though, I do think it compromises. You throw a polymer in the equation, it's like, what?
yeah, at that point, when you're dealing with something that is so specific, it's like, if you have something that's supposed to be the absolute top of its performance, top of its class, and then you have a really, cheap aspect of it that just doesn't seem fitting. But it does seem like something that almost feels like something that people just might not even normally look up or know or care about, um, which is why I really like the way you answer the question here. So, moving on to the next and final question here. So, for a biotech company that's currently using freeze drying for their biologic product, what would the case look like for switching to dynamic atomization? And when would it maybe not make sense if there is a case that it wouldn't?
Well, freeze drying can make sense. It's a fairly, I would say, simple approach, but it's not necessarily cost effective in a lot of cases. If you're doing it in house, the equipment can be expensive, but it's it's costly. The equipment is costly, and the equipment is slow. The processing through that equipment is slow. By comparison, our proprietary technology is roughly one-fifth the cost and 5 times the throughput, and the quality of the powder is the same. We get a freeze-dried quality powder, but we can do things in the creation of that powder, in the drying, that are far more versatile. So freeze drying basically just extracts the water out of the material. And therefore you're left with a powder form, but a powder that has no protection. With our technology, the dynamic atomization technology, you can do a range of things in the customization, the creation of the particles that give you a protected and coated powder. We focus on the cyclodextrin inclusion because we've perfected the ability of that cyclodextrin inclusion to wrap the individual molecules and dry them, remove the moisture around that molecule, eliminate the water activity, and then deliver that individual molecule and unwrap it, giving it tremendous shelf life. The freeze-dried powder has the benefit of adding shelf life. That is one of the benefits of the freeze-dried powder, but the extension of the shelf life with freeze drying is not great. I would say it's midterm, whereas the extension of shelf life with the cyclodextrin inclusion is long term to ultra long term. I think a company has to determine what's their goal, how much shelf life extension do they need, and then the second thing is what do they want to do with those powders. If they want to deploy those powders in multiple applications, they have to determine if they want to use those powders in applications where those powders need to be protected for a while. So, for example, the benefits of the cyclodextrin inclusion allow you to deploy those powders in the gastrointestinal tract, whereas a lyophilized powder would never survive in the gastrointestinal tract. You could never get it into and past the stomach. It would dissolve and be destroyed. If you want it to dissolve in the mouth or under the tongue, or you want to put it in a solution and have it dissolve immediately, it can do that. But it's limited to a very small number of applications, whereas a powder with the cyclodextrin inclusion go into dozens or scores of applications. And so the question is, how does it really need to be used? And so the economics around those things have to be evaluated based upon, the scale of production, how far down the channel you're going, what the applications are. But the technology allows for very substantial throughput. And if there's any scale at all, then the dynamic atomization technology, cyclodextrin inclusions, and so on can actually be very competitive financially. In many cases, much more practical financially, as well as more versatile. So lyophilization tends to be a good bet if you just want very small micro batches of things put in small vials, the way they have historically done peptides to date. If you're going to reconstitute them and you can keep them in a cold chain, or they're stable enough materials that they can survive ambient temperatures for a while, so you don't have the cost of a cold chain. But you need a relatively stable product to enhance your business case. If you have a relatively stable product that helps your business case, then it may not be a bad solution.
So I do have one final question that is really honestly out of curiosity for like myself with a lot of the questions that have been answered. So within the versatility of, I think you even just touched on it, but lyophilized or freeze-dried product can be reconstituted, but it can't really be used in terms of if you want that to be digested in the gut, maybe immediately orally, but it'll never make its way down to the digestive tract. I understand that dynamic atomization, and specifically the cyclodextrin inclusion that you guys do, can make it so that it can be used in a handful of different ways. Can that product from you also be utilized to reconstitute later for different purposes, or is it now only being able to be used internally or
Forever.
digested?
You mean is it forever powder and stays forever powder?
Yeah, basically. Does it have to essentially be, made into a powder or a solution or something that gets consumed directly orally by somebody, or can it be reconstituted as well?
Sure. It can be reconstituted as well.
Okay, that's
I
interesting.
mean, it can be, it can be powder in a glass vial that's reconstituted for injectables, IV bags. Um,
All right,
We
cool. No, that
we
was my
we
last question because I
oh,
was.
hold on. Will has an addition.
well, I was just going to say we have great examples of that now. Our powders go in sleeves to make drink mixes and so on, and we have great examples of one of our patented products, the sodium caprate, which is so highly soluble. You basically take it directly off the production line, take a spoon of it, throw it on top of a glass of water, a beaker filled with water, and it simply disperses in the water, and the water immediately turns clear. It absorbs the powder, and it's we can engineer the particles in the powder for dispersion and solubility specifically to facilitate that.
Wow. No, I asked that question because I was trying to find something that could have been a mark against that side of it, but I couldn't. I was like, well, maybe there's one thing that could be bad about it, and it doesn't seem like there is after that.
Yeah, well, again, people choose their technologies for all sorts of reasons, and in the peptide space, it's just a category that suits the technologies we're talking about very well.
No, it seems like it. Well, that is every question that I have for today, and even a couple more that came up. So, thank you guys so much for hopping on and doing this with us today. And I think we'll probably come back with more questions soon.
Fantastic.
Sounds great. Thanks, Stephen.
All right.
Thank you very much.